I lead our data and bioinformatics team at Addgene, and we're really excited to tell you about the platform that we developed for sequencing plasmids to really optimize that procedure that we have at Addgene. So Addgene is a nonprofit biorepository. We started in 2004. Partly as a result of one of our co-founders' experiences trying to request materials from other laboratories for her research in graduate school. And so Addgene was started as a plasmid repository to make it easier for scientists to share those kind of DNA-based materials. And a lot of basically, the idea behind Addgene at the beginning was really just to make it easier for scientists to share those and to be able to build on existing research much easier and faster. And since we started, our mission really hasn't changed that much. So, we want to connect scientists and to accelerate the pace of research and discovery. Since we have started, or since our initial plasmid repository started, we have expanded into other DNA-based materials that are derived from plasmids, so viral vectors and antibodies. And recently we've started a data hub where we're encouraging scientists to deposit their experimental data with us so that scientists can share what works and what doesn't work, and really make it easier for scientists then to plan their next experiments. So plasmids are just a really common tool in life sciences. They're double-stranded DNA sequences in a circular structure, and they're easily copied in bacteria. So really quick and easy to use in the lab. They range in size from a few thousand to tens of thousands of DNA bases, and they can contain repeated sequences or motifs in those sequences. Some of the common uses for plasmids include just cloning, so a handy way to copy a DNA sequence like a gene, gene expression, so expressing that protein that's encoded in that DNA sequence in a cell or a tissue. Genome engineering like CRISPR is also plasmid-based and is a way just to edit or specifically modify a DNA sequence in a cell or in an organism. And finally, plasmids can be used to make specific protein or viral tools for experiments. So the basis of Addgene's sharing model is just that when a scientist creates a plasmid in their lab and eventually they publish their work, they submit or they publish in a scientific journal, and at the same time, they deposit that plasmid with Addgene. And so what we do at Addgene is to verify that DNA sequence for everything that comes in, and this is where our bioinformatics needs originate. Then on the, I guess the next sort of part of the model is when other scientists read that publication, they can come to our website, they can find that material, and they can request it from us. It only takes a few days on average, from placing an order to getting it in their lab. So our model really saves time for everyone. The scientist who's made the plasmid doesn't have to respond to requests. They don't have to, ship things in the mail. And the scientist requesting the material can benefit from our shipping operations and the fact that we have checked all of the samples that come in. Our repository has constantly grown since we started, so now we have more than 140,000 plasmids in our freezers and available on our website for request. And we have more than 410,000 DNA sequences to go along with those plasmids. And we're constantly adding new plasmids and sequences each day. And on average, we're taking in about 1,200 new plasmids each month. So this is kind of, this is really the impetus that we had for developing a bioinformatics solution that was more automated and could help us keep pace with this intake. So this figure here just sort of simplifies, or it's a simple diagram of the sequencing steps that we take at Addgene. So there's a lot of work that our lab-based teams do in this sample prep stage, but essentially they work in big batches of things. We work or we handle at least 96 samples at a time. Again, this is just a common sort of lab format. And so when we have a batch of plasmids that's ready for sequencing, we label each of the individual plasmids with a unique barcode, and then copy a fragment of the plasmid, including that barcode, which we then, sorry about the numbering there. We then put that on our sequencing instrument, and then that reads the DNA sequence for those tiny fragments, including the barcode, and then we're able to tell which of those fragments came from which original sample. And so this kind of short read sequencing, it's nothing new, but the challenge for plasmids is that they're circular in structure, and they can contain a lot of repeats that can make the assembly part a challenge, because we have to be able to have sufficient overlap from all of those fragments to be able to recapitulate the entire sequence. All right, so zooming on what was supposed to be step 3 on that previous diagram, our assembly part. So the key parts that we needed to do here were to transfer the raw data files from the sequencing instrument to our compute environment. So initially, this was our laptops or maybe our office servers, but we wanted to put that into the cloud. And then after we've transferred those files, then we generate the FASTQ files for each of the samples, and then perform some quality trimming just to prepare those FASTQ files for the next stage, which is putting it or using an assembly algorithm to try and stitch together those little fragments. And so, there was no open source, ready to use workflow specifically for plasmid assembly. So what we did was just look at the available assemblers that had been used for genomic sequencing and test out which ones we thought might work for plasmids. And as we were doing this, we ran into some I guess, disadvantages to our initial prototype, which I'm sure will sound familiar. We had to use the command line for everything. It was one step at a time. You have to closely monitor everything. It's not easy to train others to do this. And we were initially limited by just the available compute resources that we had at the time. So here I'm just going to show a little bit more in detail the custom workflow that we developed. So the first step, as I said, was just to transfer the raw data files into our compute environment. And then we needed to generate the FASTQ files using the BCL to FASTQ program from Illumina. We then needed to perform some pre-processing or quality trimming of those FASTQ files. And we used the BB Tools package from I think it's the Joint Genome Institute, starting with BBDuck to trim out or to remove those low quality reads from our samples. We then merged some of the overlapping paired reads to make longer reads. And then the BB norm process just to even out the coverage distribution for those samples so that it was easier, well, it's, I guess, faster for the assembler, as well as it improves its success in assembling all of those fragments together. So after these pre-processing steps are completed, we've got a FASTQ file that's ready for assembly. And we had identified 3 assemblers that we thought would be useful here. So we used spades, shovel, and novoplasty. And I guess up front, we didn't know which of these was really going to be best for for a given sample. So what we decided to do was to use both spades and shovel on every sample that we had. And these assemblers generate a linear sequence. So then the next step after that was to see if we had enough overlap on either end of that linear sequence so that we could circularize it to get back to our plasmid, or our complete circular plasmid sequence. And so we used a tool called APC for that part. For any of our samples where spades and shovel both failed, we had an alternative pathway where we used this novoplasty assembler, which was originally developed for circular genomes. So not specifically plasmids, but we thought maybe it's close because it handles circular. So we try that for anything that fails the first 2 because it does run a little bit slower. but we did find it did return some useful results for when the other two failed. So then, once all of our assembly part is complete, then what we do is we select the best available sequence that was generated, and we generate a summary file so we know which assembler was used for which sample, whether it generated a linear or a circular output, and then we packaged all of the FASTA files together so that for our quality control team to then later analyze. So we had this prototype that worked, but we needed to put this into production in a way that would be sustainable for us. So we obviously needed to chain all of our scripts together so that we could move away from the command line. We wanted to put this in the cloud so that it wasn't dependent on a specific computer, or it wasn't something that only one person could do. And we also wanted it to be able to scale, right, so that we could devote more resources in case we either need a faster runtime or we have a lot more samples that come in. Being able to run things in parallel was really important to us, just given the number of samples. On average, it's like 300 to 400 per week. And the way that we had structured the assemblers, we didn't want this to take days. We want it to be completing in hours. And then we also were looking for a good user interface for our quality control team. So, something that they could, easily figure out how to use, and something that would also work for monitoring the process and seeing, if any errors occurred, what was the problem, how can we restart it. And then finally, we needed some sort of access controls because we wanted to segregate the sort of development side of things from our production workflow. And I'm sure it's no surprise here, we found Sakara, really hit all of those requirements for us and was really, ready to use for us. So to summarize, our assembly workflow really fit into our overall quality control process at Addgene. So once the samples had been prepared and sequenced, the workflow we developed in Sakara could take over, right? And this eliminated all of that manual process that we had to do at the beginning, and was saving our team hours and hours each week. But as we were doing this testing, we realized that there was more that we could do here. So this is what's indicated by the quality screening sort of branch here on the diagram. So one of the other tasks that our quality control team did was, or does each week, is to check the raw sequencing reads and look for bacterial sequences that might indicate some contamination in our stock. So this was something that one of our team members was just running manually on their laptop after the assembly process. But we realized, we could just put this into the same workflow, have this branch after we've generated the FASTQ files, and then again, just saving them time and automating more of the process so that they can spend their time, where it's really needed, and this just increases the amount or the throughput that they can do so that we have more plasmids available for scientists. All right, and then finally, I'd just like to thank the team that actually did the work. So Shogun is our data engineer, and he wrote all of our Nextflow scripts and transitioned everything to the cloud and Sakara for us. And Carrie and Zach from our quality control team helped us with the initial requirements and a lot of testing and feedback and great ideas. And so with that, I'll thank you for your attention.
Okay, we've got a couple of questions there. First up, we've got a question from Wanda. What cloud computing platform does your workflow use?
So we use AWS.
Simple.
Yeah, luckily Sakara takes care of all of that setup for us, because honestly, I'm not an expert in all of the compute environment. So we were really happy that just you can run batch forge, I think, and then set everything up for us.
There's another question there from Harshal. Would long reads be better than short reads, especially for the assembly?
Yeah, so there's a definite advantage to long read sequencing, and that's something that we're looking into. For our purposes, the advantage of the short read sequencing is really just the throughput, right? So we can do hundreds of samples all at once, and I think our quality control team is looking into sort of how to multiplex with the long read sequencing, but yeah, we haven't made that transition yet. Cool. But good question.
Okay. Awesome. Well, thanks a lot, Jason. And for the next speaker. Thank you.